So let's talk more about the cytoplasmic organelles. We're going to start with the mitochondria. It's called the powerhouse or the power plant of the cell. That's because it provides the cells ATP, and it provides this ATP via cellular respiration. And both chloroplasts in plant cells and mitochondria in animal cells and plant cells have their own DNA and RNA. It was once thought, and is still thought, that there's an endosymbiont theory wherein mitochondria and chloroplasts were once prokaryotes and have managed to make it inside of eukaryotic cells and now play a major role. There's also ribosomes. Ribosomes can be free or bound to another substance. Ribosomes, no matter where they are the site of protein synthesis. Some membrane-bound ribosomes are found on the nuclear envelope, and they're found on the rough ER, or rough endoplasmic reticulum. The endoplasmic reticulum comes in essentially two flavors. Rough ER, which is studded with ribosomes, so appears rough or bumpy under a microscope, and smooth ER, which does not have embedded ribosomes. Rough ER plays a role in protein synthesis and in the synthesis of plasma membrane proteins and phospholipids. So we have ribosomes, we have protein synthesis, so ribosomes share that function with the rough ER because of their presence on the rough ER. But the smooth ER plays no role in protein synthesis. It does contain enzymes, it helps catalyze reactions, it does have a tubule look similar to the rough ER, but is a little bit more looping. It also plays a role in lipid metabolism and absorption and synthesis and transport and detoxification. So it can help, for example, with the storage of fats or the storage and release of calcium because it has a very much developed reticulum to make or produce these products. Next, we're going to look at the essentially post office for the cell. This is the Golgi apparatus. The Golgi apparatus is generally going to be located between the ER or endoplasmic reticulum and the plasma membrane. The Golgi apparatus is going to receive products from the rough and smooth ER in vesicles and modify, maintain, concentrate, and package those proteins and lipids before they're actually sent throughout the cell or out of the cell or added to the cell membrane. And lysosomes sometimes combine with things coming into the cell. Remember that vesicles or membranous sacs or cell bubbles? Lysosomes are specialized membranous sacs that contain digestive enzymes called acid hydrolases. Now, they're considered safe sites for digestion because they isolate anything that's potentially harmful to break it down. Things that lysosomes help break down is ingested bacteria, viruses, toxins. They can also help degrade non-functional organelles. They can help break down glycogen to release simple sugars like glucose, and break down bone to release calcium. They can even destroy cells in injured or non-useful tissue. That's a process called autolysis. And peroxisomes are similar. Lysosomes and peroxisomes are both membranous sacs, but lysosomes contain digestive enzymes. Peroxisomes contain detoxifying enzymes. So they're able to break down things to the extent that they're no longer dangerous if they were to be released from peroxisomes. Our two main detoxifiers in peroxisomes are going to be oxidase and catalase. Oxidases can convert toxins to hydrogen peroxide, and catalases can convert hydrogen peroxide to harmless water. Because hydrogen peroxide free in cells or the body can actually damage DNA because it acts as a free radical. And peroxisomes help to neutralize dangerous free radicals because they're highly reactive chemicals that have unpaired electrons and can damage cell structures. So thus far we've been looking at the endomembrane system, and it consisted of membranous organelles like mitochondria, Golgi apparatus, endoplasmic reticulum, both smooth and rough, and the secretory vesicles like lysosomes and peroxisomes that are similar and mostly modified. But we can't forget the nucleus. The nuclear and plasma membranes also are a part of that endomembrane system. And these membranes and organelles work together to produce, degrade, store, and even export biological molecules to other cells or just to an interstitial space. They degrade potentially harmful substances and also keep us safe. But that's not to say that all organelles are part of this membranous system. Some do not have that membrane. like the cytoskeleton or cell skeleton, which is really just an elaborate series of proteins that kind of look like rods that go throughout the cytosol. They include microtubules, intermediate filaments, and microfilaments. Microfilaments are the thinnest or smallest of the cytoskeletal elements. They are made up of units of actin arranged in two strands and wrapped around each other. They're involved in things like cell motility, separating cells during mitosis as a part of cytokinesis, change in shape, including for both endocytosis and exocytosis. Then you have intermediate filaments, which are intermediate in size, and they're also going to be a part of desmosomes. So they help with resisting pulling forces on the cell. And then microtubules, which are the largest of these cytoskeletal elements. They're really dynamic hollow tubes made up of repeating units of tubulin, and they help the cell determine its overall shape. They also help keep track of organelles like mitochondria and lysosomes and secretory vesicles. And these types of organelles attach to microtubules, and they can actually be moved throughout the cell by motor proteins like kinesin or dynein. Microtubules are even a part of major functions like mitosis or cell division. And when they function in cell division, they're called spindle fibers. But not to overlook the motor molecules, these are protein complexes that function in motility. They're powered by ATP, and they work by changing shape and work kind of like train engines and use microtubules as train tracks. What makes microtubules, though, since they're so important? Well, microtubules are made by centrioles. Centrioles are found in an area close to the nucleus called the centrosome. There's generally two centrioles in a non-dividing cell. So again, the centrioles in the centrosome make the microtubules. But centrioles also form the basis of structure for cilia and flagella. And cilia are shorter than a flagella. Cilia are kind of short, motile hairs that kind of whip back and forth, and they help move things across a cell. In eukaryotic cells, cilia are not used for cell movement. A flagellum is much longer and can be used to propel a whole cell. In eukaryotic cells, it's not common to see a flagellum, but they are much common in simpler prokaryotic cells. And then you also have another type of cellular extension, microvilli. But microvilli are not something that's attached to the cell membrane, like cilia and a flagellum. Instead, microvilli are actually finger-like extensions of the plasma membrane that increase surface area for absorption. They're really just extensions of the plasma membrane, so they're very much connected to the plasma membrane, not separate and connected, but a part of. And then one of the last organelles we're going to be looking at is the nucleus. The nucleus is usually the control center in the center of the cell, and cells can have one nucleus, multiple, none. If they have multiple, it's multinucleate, like skeletal muscle cells. Just one, that might be something like a smooth muscle cell or a neuron. And other cells, like red blood cells, get rid of their nucleus, and they don't last very long because of that, but they do that for functional regions. So just keep that in mind when we're talking about a nucleus. There can be more than one in a cell, and there's generally three regions of a nucleus: a nuclear envelope that contains nuclear pores, the chromatin, and the nucleolus. So we're going to start with the chromatin in the next video.